to start off by saying thanks for the organizers. Thanks to the organizers for inviting me. My name is Corey Susky. I'm an associate professor here at the University of Illinois in Urbana-Champaign, Illinois. And I really appreciate the opportunity to be here and share with everyone some of the work that we've been doing related to barriers and stopping the movement of Asian carp. So thanks very much for inviting me today. Before I dived, this is some research. The point of the talk really is to cover some of the research my lab group has been doing here for the past 3 or 4 years or so. But before I dive into the specifics of what we've been doing and what we've been finding, I wanted to start off with a little bit of background information to kind of set the stage for where we're going and how some of this work really fits in. And ultimately, the talk is going to be, and the subject is about Asian carp, which are an invasive fish species. And so if you sort of take a step back and look at invasive species broadly, not just fish and not just Asian carp, what you see is that invasive species, especially aquatic invasive species, are very common around the world. There's a number of sort of high-profile cases of invasive species in a number of different locations around the planet. A lot of the reason why invasive species are so prominent and so abundant is partially fueled by sort of global trade and commerce. I saw a paper a few years ago I remember reading about was just the number of international flights that moved around the planet and the number of people sort of moving around the globe on a daily basis, and it was pretty scary. So our ability to move around the planet is actually really, really easy. Our capacity for global trade, global commerce, container ships like the one I'm showing here in the slides, very common, very prominent, and so it's really, really easy for a lot of these invasive species to kind of hitchhike and get kind of dragged around. The reality is that if you do have an invasive species problem, if you do have an invasive species show up in an area, it's much easier to prevent, or it's much easier to prevent that invasive species from showing up in the first place than it is to try and remove the invasive species after it's shown up. So prevention is the key when it comes to invasive species, and I really want to kind of emphasize that point as the talk kind of moves forward. When we talk about some of the impacts of invasive species, I'm here in Champaign, which is quite close to the Great Lakes area, and the Great Lakes are kind of been the poster child for invasive species and the arrival of invasive species, specifically aquatic invasive species. And this is a paper that came out a few years ago now that shows the cumulative number of invasive species arriving in the Great Lakes Basin over the past, say, 150 years or so. And so the vertical axis or the y-axis is the cumulative number of invaders. And what you see is that there's been, creeping up on 200 invasive species that have shown up in the Great Lakes over the past 100 and so years. Those animals don't all establish, they don't all start spreading, but animals are showing up in the Great Lakes Basin over the past little while to the tune of about every 28 weeks something new shows up. So the Great Lakes are particularly vulnerable and particularly susceptible to a lot of these invasions, and they're kind of the hot spot for a lot of stuff that's been going on. When we talk about why we care about invasive species, one of the easy ways to kind of make people take notice of invasive species when we start talking about dollars and start talking about costs. And there's a lot of information out there on this. For example, introduced fishes are believed to result in economic losses of $1 to $5 billion annually. There's a huge sea lamprey removal program going on in Canada and the US to get rid of sea lampreys and their impact on fisheries. And billions are spent on invasive mollusks, zebra mussels and quagga mussels and those kinds of things in terms of clogging water intake pipes and those kinds of things. So huge problems, and it's really easy for people to kind of grasp and get on board when we start talking about costs and the billions of dollars in costs that invasive species have. The reality, though, is that there's a number of costs beyond just dollars to invasive species, and especially when it comes to diversity and biodiversity, which is the topic of part of the reason why we're talking here today. And when you kind of look at sort of the literature and some of the information that's out there on these topics, is you see that invasive species can displace native species, they can alter ecosystem processes, they can change community structures, and so when you have an invasive species arriving in an area, it can have huge impacts on the receiving environment. And broadly, freshwater ecosystems may be among the most threatened ecosystems in the world for a number of reasons: habitat loss, destruction, invasive species, climate change, all these kinds of things. But the figure that we have there in yellow is sort of like biodiversity on the sort of vertical axis over time, and we see that the purple line is biodiversity and species indexes for freshwater, and you see that we're losing species from freshwater faster than we are from terrestrial or marine environments, and freshwater ecosystems are very, very threatened, so they're very sensitive. And again, just to sort of reiterate that point that I made earlier, keeping invasive species away is a much better strategy for protecting biodiversity and local ecosystems than it is to try and remove those species afterwards. So prevention here is the key that I'm trying to emphasize. So this is invasive species in freshwater, broadly defined freshwater ecosystems. But when we don't talk about freshwater fish are incredibly sensitive still, or much more sensitive still relative to some of the stuff that I talked about before. Across the paper came out a few years ago, across North America, 39% of fish are currently imperiled or threatened. So a third of all the freshwater fish in North America have some sort of conservation problem. And the top paper that I'm showing there on the figure came out in 2008, and that was an update of an earlier sort of survey that was done in 1989. So from the period from 1989 to 2008, what you see is there was a 92% increase in the number of imperiled fishes in North America. So over time, things seem to be getting worse for freshwater fish. So things are bad, things seem to be getting worse. And when you go back to the literature, extinction rates of freshwater fishes are believed to exceed that of terrestrial fauna. So fresh waters in general are suffering a lot of losses in biodiversity, and then freshwater fishes, again, are losing a lot of biodiversity as well. So very, very sensitive taxa and a lot of conservation problems, sort of broadly defined. And so with that as sort of the doom and gloom background opening to a lot of the stuff I'm going to talk about today, I can move into things that are a little bit more specific now. We can talk about Asian carp. And specifically as it relates to this talk, you can't really talk about Asian carp without talking about the city of Chicago and the Chicago area waterway. And so those are the sort of the two players that I'm going to talk about in the story today. And so as we move forward, it's important to kind of keep both of those aspects in mind, not only the biology and the fisheries side of things, the Asian carp, but then also the Chicago area waterway system, which sort of represents sort of the economics or the policy sort of side to the story as well. So the carp side of the story is actually relatively easy to tell. It's relatively straightforward and something that people can a lot of people sort of know about possibly already. In the 1970s, Asian carp came from Asia and were brought to the U.S. for aquaculture purposes. Asian carp are they're filter feeders, and so the best way to kind of describe how they function is that Asian carp, they eat when they breathe. So as they swim around a body of water, they're breathing, they're drawing in water into their mouths, and anything in the water, they're basically kind of filtering out, and they're actually eating that. So they're very good at filtering water and keeping water clean and filtering out small zooplankton and phytoplankton and those types of small organisms out of the water. So they're great for aquaculture. If you have a fish farm or a fish pond, Asian carp would be, in theory, the reason they're brought over is they're great to keep that water clean and keep the water filtered while you're growing other things like, say, catfish or other sort of commercial fish. But then what happened is in the 1970s there was a flood. They're in Arkansas, I believe. In the 1970s there was a flood, and the Mississippi River encroached onto that fish farm, and the Asian carp got out. And since then they've been spreading north, and they've been spreading south ever since they were released sort of in the 1970s. So that's how they got here and how they came to spread around past where they were originally introduced. Why do we care about Asian carp? Well, they spread rapidly. They're very abundant. They're very fast growing. They're highly fecund. A female can have between 265,000 and 2 million eggs, and they can have multiple spawning bouts per year. They compete with native species for food. So every small fish, every small fish in a river or in a lake, when they're very, very young, say the size of your finger, they will all compete. They will all eat small phytoplankton and zooplankton. So every small fish competes with what adult Asian carp eat. So they have a lot of they compete for food with a lot of those native fishes. Everyone's seen the videos, hopefully, or the pictures. If you haven't, you can go type that into Google and look at some of the videos of jumping carp or Asian carp. They jump out of the water, which can be very dangerous. They can hurt people. So they can have negative impacts on recreational fisheries and then on non-sport fish as well. So very serious problem. And a lot of stretches of the Illinois River in particular, Asian carp are now the most abundant fish in the river. So they spread rapidly. They become very, very large in terms of population size, and they're very large in terms of individuals. Not a good thing to have around in a river, and very difficult to get rid of once they do show up. So that sort of is the fish side of things, and we can talk again about the other player, which is sort of the Chicago area waterway. And I'm not exactly sure where all the folks on the internet are from, but for those of you that might not be from Chicago or even from North America, the city of Chicago is sort of located around the center of the United States, incorporated as a city in 1837. It's the third largest city in the U.S. It's got the world's second busiest airport, and it's a major hub for finance, tourism, and industry within the United States. So it's a very big city, very populated city, and a lot of things going on in Chicago. So it's kind of a big hub for a lot of things within North America. Where the story then becomes a little bit more specific is when we talk about something called the Chicago Area Waterway, or the Chicago and Sanitary and Ship Canal. And back in the way the story goes is that back in sort of in the late 1800s, early 1900s, Chicago was using Lake Michigan to both obtain its drinking water and dispose of its waste, which is not a really good practice if you're trying to keep people healthy. And so they decided they needed to change how they were specifically dealing with their wastewater, and come up with a new solution to getting rid of their wastewater. And so what we have actually right around the center of the city of Chicago, or the Chicagoland area, is really, really interesting, because what there essentially is, and I have a picture of this in the slide in the top picture, there's basically a continental divide, in that there's a sort of, on the top picture, there's a vertical dotted yellow line, and that represents a continental divide. And anything to the left of that vertical dotted yellow line represents the Mississippi Basin, and anything to the right of it represents the Great Lakes Basin. And so literally the Great Lakes Basin and the Mississippi Basin are separate basins. So the water on the left-hand side of that vertical yellow line all drains into the Mississippi. The water on the right all drains into the Great Lakes, and they were totally separate waterways, or totally separate basins back in the late 1800s. But again, Chicago, there's a need to better deal with sewage, to better deal with wastewater, stormwater, and those types of issues. And so Chicago built 78 miles of canals to help aid in that removal of wastewater. And those canals were artificial connections, again, that sort of ditches, essentially large canals that we dug as humans, and dug across that continental divide. And you can see the new map on the bottom there, where now we've got rivers that used to flow north are now running south. There's been pumping stations installed on the shores of Lake Michigan, and Lake Michigan water now is pumped into the rivers, through the canals, across that continental divide, and down basically into the Mississippi Basin. It's actually the Great Des Plaines River, but that flows into the Illinois, it flows into the Mississippi. So what the point is, we have now artificially crossed that continental divide, and are sending Lake Michigan water down through the Mississippi Basin, where 100 plus years ago it didn't actually happen, and those were separate basins, and those were separate drainages. So the present, then, what we have is the Chicago Area Waterway. There's a couple of pictures of it here, the spectacular waterway. It receives stormwater, so rainwater overflows from 740 square miles of urban areas. It contains treated sewage discharge. There's a very large shipping and transport industry that uses that to transport goods and services up and down the Mississippi, to and from the Great Lakes, up and down to the Mississippi, say down to New Orleans. There's recreation that happens in terms of boating and fishing, but at its core, what it is, it's an artificial open connection between the Mississippi Basin and Great Lakes Basin that allows the passage of aquatic species. So previously, prior to humans, there was no way that animals could swim from the Mississippi Basin into the Great Lakes Basin, but now that we are here, now we made these changes, animals can swim from the Mississippi Basin to the Great Lakes Basin, specifically Asian carp. And the thing we're worried about is that Asian carp have the potential now, because of these canals in Chicago, and a few other different points as well. The Chicago one's a more high-profile one. Asian carp can now swim from the Mississippi Basin into the Great Lakes Basin, potentially. And so that's got a lot of people nervous that the animals, should Asian carp pass from the Mississippi Basin into the Great Lakes Basin, then in theory, a lot of Eastern North America is open, because you can swim through the Great Lakes, then you're into the St. Lawrence River, and you can distribute around a lot of Eastern North America. So it opens up a lot of new territory for the animals that they previously did not have access to. At present, there's a lot of work going on to control populations and harvest and removal and these kinds of things. And in addition to that, there's one electric fence, an electric barrier that's in Romeoville, Illinois, and designed to stop the passage of fish. Okay, so literally it's an electric fence. The water can still flow downstream, but there's an electric fence there that in theory could stop Asian carp from moving through the canals. The reality too is that there's, the Asian carp at present are likely still about 30 plus miles away from Lake Michigan, so they're not currently, knocking on the door of Lake Michigan, and that canals, the electric fence is there to stop their movement. Information to date says that the canal's probably been effective, the electric fence has probably been effective. Asian carp likely have not swum through that electric fence, but the reality is that when you have a non-physical barrier like an electric fence, no non-physical barrier is going to be 100% effective. Electric fences are prone to things like power outages, routine maintenance. There may be interactions between fish and barges moving through that may provide the barge may provide cover or protection from the electricity, and electricity in general may, these types of barriers may start to lose effectiveness against small fish. So the point being is that this electric fence, while believed to be effective to date, it's not going to be 100% perfect, and it might not be the best solution, especially over a long term. So now we need to figure out what we can do long term to stop these animals from moving from the Mississippi Basin into the Great Lakes. And the Army Corps of Engineers put something out called the GLIMRIS report, and GLIMRIS is an acronym that stands for the Great Lakes Mississippi River Interbasin Study, and that's shortened down to GLIMRIS. And you can go look this up on the internet. It's a big study. And basically what this study does is it says, what are the options that are potentially available to stop aquatic species from moving through Chicago from the Great Lakes Basin into the Mississippi Basin? What are the options? So it's not going to tell us what to do, but it'll tell us, like, here's a bunch of options for people to ponder. Everything from doing nothing to, physical separation. And so there's a lot of options involved. It's a very comprehensive, very thorough document. It's very, very interesting. And I'm not going to go into a lot of it detail, but and you can go download it if you want and check it out. But the point is, I cherry-picked out one of the potential mitigation measures that the GLIMRIS report talks about, and there's a picture of it here. There's a picture of Chicago. A bunch of different barriers happening, changing of locks, building reservoirs, all very extensive, but it's very, very, very effective. The reason I bring it up is because on the bottom right-hand corner of the slide, you can kind of see the price tag for that, and this particular barrier option, or this particular mitigation option, is running in the ballpark of $18 billion. I don't have the timeline in there, but it'd be 20 plus years to actually build and implement. So we're looking at, for these kinds of things, very, very long-term solutions, very, very expensive, effective, but long-term and expensive, and someone's going to have to come up with $18 billion to cover these kinds of things. So for the short term then, what we're looking for is what can we do to help the electric fence that's in Chicago while we start to figure out, longer term issues, longer term solutions, and how can we basically help improve the likelihood that we can stop Asian carp from moving from the Mississippi Basin into the Great Lakes. And so now we start talking about some of the stuff that my group has been doing, which is starting to use carbon dioxide as a non-physical barrier. And if you go and talk about fish barriers, there's basically two different kinds of fish barriers. There's what's called a pull barrier or an attractor, something that brings fish into an area, like a pheromone or a hormone, and fish want to be there. Or you can develop a push or distractor barrier, which keeps fish out of an area. For example, a bright light or a loud noise, and the fish kind of get startled and swim away. We've kind of gone through the literature, and the reality is that for a lot of these non-physical barriers, nothing's going to be 100% effective. Like, fish are going to kind of, it's likely that fish are going to find a way through. So each one thing by itself isn't going to be perfect. But if you can start to string together multiple non-physical barriers, you can maybe increase redundancy, increase safety, and increase the likelihood that you can stop fish moving. And for a lot of reasons, it's really interesting to look at small fish and the ability to stop small fish. So basically what the project that we were doing is looking to do is, what can we add to the water to deter fish from moving into an area? Ideally something non-physical that can keep water flowing, keep barge traffic moving, and keep those kinds of things happy within the city of Chicago or elsewhere. And one of the things we started looking at then for trial and error, and we kind of stumbled on things after a little while, was carbon dioxide gas, or CO2, the same thing that's in soda, carbon dioxide gas. And the reality is that it's got potential to act as a non-physical fish barrier. Carbon dioxide gas is a naturally occurring compound. When you add carbon dioxide gas to water, it forms carbonic acid, which is a weak acid. So we're not talking about things like acid rain, where we have super, super low pH, which is really, really acidic. It's a weak acid, so it's a 1 to 1.5 pH point drop when you form carbonic acid. The ability to sense carbon dioxide in the environment is common across taxa. So fish have CO2 sensors in their gills. Plants can sense carbon dioxide. Bacteria can sense carbon dioxide. So it's very, very common across taxa. And again, fish have carbon dioxide sensors in their gills, and they even have internal CO2 sensors. So it's got a lot of really good potential in terms of acting as a non-physical barrier. So now to transition to some of the data that I have and some of the work that my lab group has been going, the goal of the talk then, and hopefully what I want to talk about for the rest of the presentation, is to define the efficiency of carbon dioxide gas, or CO2, to act as a non-physical barrier to influence fish movement. Can we use carbon dioxide gas to stop fish from moving? Is what it comes down to. And then this again would have implications for invasive species control of Asian carp, which then in turn has implications for biodiversity and minimizing the impacts of invasive fishes on receiving environments. And again, this is coming down to prevention, right? Prevention of fish from moving, because it's easier to prevent animals from moving than it is to try and remove them afterwards. And so can we use carbon dioxide gas to achieve this goal? So talk is going to have 3 sections. First section is going to be just talking about the responses of fish to carbon dioxide. What happens if you put fish in high carbon dioxide? The second section is going to talk about avoidance and about fish avoiding carbon dioxide. And the third section is going to be on what's called acclimation. And basically, can fish get used to carbon dioxide? So can they acclimatize or acclimate, and can they get used to it? So we start with the first section, which is what we're calling the physiological responses to carbon dioxide. And basically what we're asking with this one is, how do fish respond following exposure to elevated CO2? What happens to a fish if you put it into high carbon dioxide? So the goal is to quantify impacts of CO2 exposure on larval fish and juveniles. And so the top picture, we'll start with the bottom picture, which is a juvenile silver carp. So that fish is, as you can see from the ruler down there, it's about 7 centimeters long. So we're looking at something that's probably about the size of your finger. It's a very small fish. The top picture actually is a plastic drinking cup with some window screen on the side of it. And when you may or may not be able to see inside the slide, depending on what's on your screen, there's actually a little baby fish. So there's 8-day-old carp larvae in that cup. So the animals are about the size of your eyelash. And so there's a whole bunch of them in there. And so we're working with 2 sizes of small fish, about the size of your finger and then about the size of your eyelash. We got larvae that are 8 days old that are 7 millimeters, and we get those 2 to 4 inch larvae. Basically what we did is expose them to carbon dioxide. And when you're dealing with animals this small, we're looking at just stress, which is a sort of a really broad, squishy term, but it's like it's physiology. It's like if you go to the doctor and if you're sick, the doctor can take like a blood sample and tell a lot about how you're feeling based on blood and based on internal changes inside you, and that's what we're doing here with these animals. And one of the things we look at is genes and how genes are changing following carbon dioxide exposure. So basically, again, what we did was we took animals, put them in high carbon dioxide, left them in there for a little while, and then we sampled them for a bunch of different tissues. And so this figure here shows the vertical axis, or the y-axis on this one, is a compound. It's HSP70, so it's a heat shock protein. And heat shock proteins are produced when proteins inside your body start to break down. So a lot of your body is protein, and when proteins start to break down, your body says, whoa, I got proteins breaking down. So it produces something called heat shock proteins, which come out and it tries to help your own body. So when you have increased levels of heat shock proteins in any condition, it usually is a sign that things inside your body are breaking down and things aren't really going well. So that's the response that we're measuring here. We have the 8-day-old carp larvae, and we have the black bars are animals that have been sort of just left in control, just resting conditions with no carbon dioxide. The blue bars are animals that have been in 70 milligrams per liter carbon dioxide, when the actual number is not important. It's sort of a low concentration of carbon dioxide. And then the red bar is animals that have been in 120 milligrams per liter carbon dioxide, which is kind of a high concentration. The grouping on the left is animals that have been in carbon dioxide for 30 minutes, and then animals on the right, the groupings on the right are animals that have been in the groupings in high carbon dioxide for 60 minutes. And so these are the Asian carp fries. These are 8-day-old animals, a little bit bigger than your eyelash. And what you see is if you put 8-day-old silver carp into carbon dioxide 70 or 120 milligrams per liter for 30 minutes, for 60 minutes, we have an upregulation of heat shock protein 70 gene. So something's breaking down inside the animal. So it's a very sort of broad marker, but it basically says the animals don't like being in high carbon dioxide. They don't like it. Things are breaking down. They're having to induce or upregulate protective mechanisms within their own body. So it just sort of says to us, yeah, animals don't like carbon dioxide, which is good. Now we can move on to juvenile silver carp, rather than so these are the ones that are about the size of your finger. And this is a gene called CFOS, CFOS turns on other genes, and so it basically turns on a lot of other protective mechanisms. So it's a great marker. Again, we got red is control, blue is sort of that low carbon dioxide, and the sort of light blue is the high carbon dioxide. Animals that have been in high CO2, or animals that have been treated for 30 minutes, animals that have been treated for an hour. And again, upregulation of a lot of the CFOS genes in the blue bars, meaning CFOS genes are turned on. Something's not right. Animals don't really like it. And even animals that are 8 days old can start to show a response saying, yeah, this is something internally is not happy with the animals, which is great, but doesn't really help us if we're trying to stop carp from swimming between basins. So section 1B then is where I talk about some of the behavioral responses to carbon dioxide. So looking at behavior. So the effects of extended carbon dioxide behavior or exposure, sorry, on fish behavior. And so what we did here is we did silver carp, and there's two species of carp right now that are sort of really of interest: the silver carp and bighead carp. The picture on the left, there's a picture of a silver carp. And basically what we did is we brought animals into the lab, we put them in those buckets like you sort of see there. The bottom right picture is just a tank of carbon dioxide that you can get from the gas supply company, and we just put animals in those tanks, bubbled in carbon dioxide to 3 different levels, held the animals for 3 hours, and we monitored their behavior during that 3-hour exposure. So literally holding fish at high CO2 concentrations for 3 hours and just monitoring behavior. And the data for this looks something like this, where what we have on the y-axis here is what we're calling irregular behaviors, so that whether or not animals are exhibiting irregular behaviors, which is things like breathing at the surface when they're, rather than sitting in the water, they go up to the surface and start to gulp air, twitching, trying to jump out of the tank, and just doing things that aren't really normal. It would suggest that they're not really happy. So what we have then on the y-axis, the vertical axis, is the proportion of irregular behaviors. The cluster on the left is animals held at control or low carbon dioxide. Then we got animals at 30 milligrams per liter carbon dioxide, and then we have 70 milligrams per liter carbon dioxide. Black bars are largemouth bass, light blue bars are bluegill, red bars are silver carp, and then the dark blue bars are bighead carp. And what you see is that as you increase the concentration of carbon dioxide, the animals start to show irregular behaviors. Again, it just sort of emphasizes the stuff I showed previously. Animals don't like being in those high CO2 environments. They start to all show this twitching, breathing at the surface, trying to jump out of the tank. Animals don't like carbon dioxide, which is a good thing if you're trying to build a barrier. The other thing we did then was we looked at ventilation rates. So the y-axis here is ventilation rate. So it's literally how fast are the animals breathing. Okay, so we're counting their gill openings, the rate. So it's ventilations per minute, so the rate at which their gills are opening and closing. These are the data just for silver carp. And so what this is then is holding animals for 3 hours. Okay, so the time is on the x-axis there. Dark blue bars are control animals that with no CO2. The light blue bars are animals at 30 milligrams per liter CO2, and then the red bars are animals that are held at 70 milligrams per liter CO2. And the numbers aren't really the same, but basically the light blue is low CO2 and the red's high CO2. So let's first look at the dark blue bars, which is ventilations per minute. And overall, everything's around, with those dark blue bars over the 3 hours, it's all basically around 50 ventilations per minute. And the point there is that if you just take silver carp, put them in a bucket for 3 hours, their ventilation rates really don't change. They don't care about being in a bucket in terms of ventilation rates. If we put animals, go to the light blue bars now, if we look at ventilation rates, again, we have them at 30 milligrams per liter. Again, there's really no change in ventilation rate. So that concentration doesn't really change animals. But if we look at the red bars, we see that we have ventilation rates for the control, and then we get to that half-hour time period, and the red bars disappear. So this shows us a bunch of things. One, obviously the animals, basically what happens is the animals they all passed out. Carbon dioxide is actually a fish anesthetic, and they went to sleep. They happily just sort of rolled over in their tanks. We pulled them out of the tanks, put them in fresh water. They all righted themselves. They all came back, and they were all fine. But the point here is that carbon dioxide is a fish anesthetic. It causes equilibrium loss, and again, it just sort of reinforces the stuff that I've shown up to this point of the data, that fish don't like being in carbon dioxide. It's a very powerful compound. It's a very powerful chemical that has strong negative consequences for fish. They just don't like being in it. Leave them in there long enough, they will go to sleep. They'll come back. They'll wake up, but they go to sleep. Very powerful compound. So to summarize sort of the first section then, Asian carp respond negatively to carbon dioxide. Okay, larvae and fingerling fishes activate their stress response after CO2 exposure. And again, I didn't have time to show all of the data. We have a bunch of different genes, a bunch of different tissues, and sort of that stress response go up. Even if you're only 8 days old, about the size of an eyelash, you still respond negatively to being in carbon dioxide. If you leave animals in carbon dioxide long enough, we start to see irregular behaviors and then ultimately leading to equilibrium loss. Carbon dioxide is an anesthetic. Fish don't like being in it, and it gives us, makes us like that in terms of designing a barrier. But the reality is, we're looking to stop animals from moving. We don't want them to just have an upregulated stress response or show irregular behaviors. We want to stop animals from moving from one basin to another. So this is all the data that I've shown so far is good, makes us think that fish don't like carbon dioxide, but what we're really interested in is avoidance. So the second part of my talk then, I'm going to talk about some work we've done where we're looking at avoidance. And first of all, I want to talk about avoidance work that we've done with animals in a lab in a controlled environment. And we want to, so in some ways too, we want to show avoidance thresholds. So what do we have to reach a critical point of carbon dioxide before animals are unhappy or they swim away, or what does it mean? So to do this work, we had something called, we use what we're calling a shuttle box, which I have pictures here on the right-hand side of the slides, and it's a computer-controlled choice arena. Okay. So what we have, there's two tanks separated by a narrow neck, and the two tank, we can control water parameters independently on each side, in each of those tanks on each side, and animals are free to move between the two tanks whenever they feel like it. So the top picture, the bottom picture is the actual picture of the setup and of the equipment and everything, and the top picture is sort of a top-down view. And just for illustration purposes, what's happened is the left side of the tank has got yellow water in it, and the right side has got pink water, and the two sides don't mix. And we can control yellow, pink on either side, and we can see the fish, and we can watch fish behavior, and we can see when fish move. And we can control water parameters independently on each side and watch what fish do. So it's a perfect way to test how fish respond to high CO2. So what we did for the experiment was we took fish, we put them in one side of the tank, and then we just slowly over time started to increase carbon dioxide. Eventually, the carbon dioxide got so high, the animal then chose to move to the other side of the tank. The initial side of the tank, we stripped all the CO2 off, brought it back down to ambient, and we started increasing CO2 in the side to which the animal moved. Once it moved again, it changed sides. We took all the CO2 off. Then we started adding to the second side. So basically what we're doing is ping-ponging fish back and forth between the two tanks. And every time they move, we just wrote down the concentration when it decided to move. We can repeat this multiple times for individuals, and then get multiple individuals, and then we can get multiple species. And we get a really good idea of what concentration animals don't like and if they're actually able to swim away. And so the data looked like this, where the y-axis here is carbon dioxide concentration. So the vertical axis is carbon dioxide concentration when animals chose to swim away. And then we got data for largemouth bass, bluegill, and then silver carp. We didn't actually do bighead carp here. But the point is that once carbon dioxide concentrations reach around 100, 120, 130 milligrams per liter, fish will voluntarily choose to swim away. They just say, enough's enough. I don't like this. I'm leaving. Which is really, now we're getting to the stuff that's really, really good for designing and building a barrier. We get animals to leave an area, which is great. One of the problems or challenges with these data, as good as they are, is that these were adult fish. Okay, so these were big. So these were, these were big fish. These were adults. Again, when we talk about electric barriers and building a new barrier for fish and all this kind of stuff, it's really nice to have data on small fish. Excuse me. So we repeated this with some fingerlings. So these are fish that have been about the size of your finger. So again, the y-axis here is carbon dioxide, and then we got bluegill, largemouth bass, and silver carp and bighead carp, the two carp species that we're interested in. And again, no differences across species. Here you got to get up to around 150, 200 milligrams per liter. But even fish that are 2 to 4 inches long will choose to leave an area once the carbon dioxide concentrations get to be sufficiently high, which is great. So now we're starting to think about, if we're thinking about barriers, we're thinking about stopping fish movements. Now we get really good data showing, yeah, this is great. The animals don't seem to like it. And then we'll swim away from it. The problem now is that when you look at these data, they're all generated in a lab, in a really small, controlled, in a shuttle box, in a controlled, small environment that might not be realistic or representative of what's happening out in the field. Okay, so what we really wanted to do now was take this to the next step and get it into a larger context, get into a big setting, and start to look at how things are happening in a more grand scale. So the final steps, the final step in this, some of the progression of experiments here was to look at use a pond setting. Okay, so everything we've done so far has been done in a controlled lab environment. What we want to do is quantify the effectiveness of a zone of elevated CO2 to influence the movement of free-swimming Asian carp, but in a pond, large scale, not in a lab, okay? Because if we are going to develop a barrier that might go, say, into like a river, into a waterway, into a canal, we need to sort of scale up to this size and do something at this scale. So we really want to get big with the experiments, with the data. So for this, we collaborated with some folks at the United States Geological Survey in La Crosse, Wisconsin. They have some ponds that we're able to use. These are half-acre ponds, 5 million liters, so a little over 1 million gallons. So we're talking, I have pictures of it coming up. So we're talking big scale, 1 million plus gallons of water. Everything we've done before was done basically with, like I showed, a tank of carbon dioxide and basically like an air stone. But now we need to change technology to go up to this size of an experiment. using something called a Venturi tube. That's actually not really important, but the point is now what we're doing is we're taking our pond, pull water from the pond, treat it with carbon dioxide, return it to the pond in the same area, and generate a zone of high CO2 in part of a pond where the water is actually treated externally. We're not just putting in bubbles here. We're not looking at, we're not trying to assess, we're not building a bubble curtain. We're not building a bubble barrier. We're not looking at the, we're not really interested in the low pH. We're actually trying to put high concentrations of carbon dioxide gas into the water to stop the animals from moving. We did that at one end of a pond. We monitored pH in the entire pond. You can use pH as a surrogate or as a proxy for carbon dioxide, but we measured in the entire pond for the study. What we did then is we had 10 silver carp and 10 bighead carp that we tagged. We used what's called telemetry, so we can basically monitor the fish positions in real time in 3 dimensions remotely. We did some native fish as well. I'm not really going to have time to talk about those today, but we're looking, there's a picture of the fish that's got that basically a telemetry tag on its back, and so it's just a positional tag that allows us to see where the fish is. The experiment went, we put the animals into the pond, gave them basically 22 hours to sort of get used to the pond and being in the pond and kind of swimming around. After 22 hours, we started adding carbon dioxide to one end of the pond. We raised carbon dioxide within the pond. We left carbon dioxide raised in that pond for about 6 hours. Inside was called a discharge manifold that I'll show, but basically 24 hours of high CO2 in one part of the pond. Then we turned the carbon dioxide off, and we continued to monitor fish movements for about 6 hours afterwards. And again, we're monitoring fish movement with telemetry, meaning we can monitor fish positions every second and a half in 3 dimensions, basically in real time. After we did this experiment, we drained the pond, pulled all the fish out, put new fish in, and repeated. So we actually have 2 trials that we're going to do here. The way the data I'm showing you, we only have 1 trial that I'm going to show you, but the data were almost identical for the second trial, so we have replication on the trials. Here's a picture of the pond that we're using. So again, this is about 1.5 million gallons. So the top left, the pond's a rectangle, and the top left picture is looking down the long axis of the pond, so you get a sense of scale and how big things are. The white posts that are sticking up, that's our CO2 infusion system. Okay, so that's just plastic pipes with nozzles. I have another picture of it later on, but that's where the CO2 rich water was basically pumped into one side of the pond. The top right picture is just kind of looking at things from a different angle, sort of across the pond. And then the bottom picture across the whole bottom is what the pond looks like when it was full. So that's when it's full of water and ready to add fish. This picture, top left, is our carbon dioxide infusion apparatus. Again, we're looking at the top right picture is a pump. So again, what we're doing is pulling water from the one end of the pond with that pump, passing it through our carbon dioxide infusion mechanism and returning it to the pond through that infusion apparatus. And we're adding, bottom left picture then is large tanks of carbon dioxide gas. Again, the top left picture is looking down the long axis of the pond. Sort of, you can see the CO2 infusion apparatus there and the white posts. Top right picture is again that CO2 infusion apparatus. In that top right picture, you can see a trailer where we did all our data analysis for a sense of scale. And then the bottom picture there is CO2 rich water being pumped into the pond. So obviously we had the pond drained down for that one, but that's what it looks like. So again, we're not building a bubble curtain. We're not building a bubble fence. We're not looking at hypoxia. We're looking at literally taking CO2 rich water and pumping high CO2 rich water into the pond, into that area where those sort of white posts are in terms of having a zone of high CO2. Data for this experiment looks something like this. So what we see here, this black square, is the footprint of the pond. That's the outline of the pond, okay? And at the bottom of that black square, those black dots, that's the CO2 infusion apparatus. So that's where we are adding high carbon dioxide gas to the pond down there. The colored things are what's called a heat map, and the different colors correspond to different fish densities. So what we have, wherever there's purple, we have high fish density, and then it transitions to blue to green to yellow and then to red, and that's the lowest fish density. So white means no fish, red means low fish density, purple means high fish density. So what you see, this panel here shows the distribution of fish 11 hours before we started adding carbon dioxide to the pond. And what you see is that the majority of fish, the highest fish densities were towards the top of the pond, a little bit in the middle, but a lot of fish hanging out down around that CO2 infusion zone where all those black dots are. They like to be down. It's a little bit darker down there. They like being around all those plastic pipes. So that was their preferred area 11 hours before we turned on the carbon dioxide. We then turned on the carbon dioxide and let the carbon dioxide go into the pond. 11 hours after we turned on the carbon dioxide, the middle panel now shows the distribution of fish. So what we've done is now what you see is it's all white around those black dots. So around the carbon dioxide infusion dots, there are no fish, and all the fish now have moved to the middle of the pond. So we're able to, that zone of high CO2, we were able to kick fish out of that area, and they chose to not swim in the area with all the carbon dioxide. 11 hours afterwards, then what ended up happening was the whole pond became a big soup of carbon dioxide because the carbon dioxide plumes spread and they were all over the place. By 20 hours after CO2 addition, we had fish swimming everywhere too. But the point here is when you look at that middle panel and the change in distribution from pre-CO2 to post-CO2, and what you see is that animals chose to swim to different areas of the pond, despite the fact that we're looking at, half an acre, 1.5 million gallons of water. We're able to keep animals out of a particular area. So the avoidance section of the talk then, to kind of wrap that up, free swimming fish avoid areas of elevated carbon. Section of the talk then, to kind of wrap that up, free swimming fish avoid areas of elevated carbon dioxide. Once you reach a target threshold, which is, and we'll talk about that a little bit later on, we replicated this now at 2 scales, both in the lab and in a big pond like that. I did not have time to go through some of the other species that we ran, but we ran a lot of native species, largemouth bass, bluegill, there were some yellow perch, all kinds of native species. They all responded in the same way, meaning that the carbon dioxide response does not appear to be species specific. Every fish seems to respond about the same way. And again, this really starts now to provide some support for using carbon dioxide as a non-physical barrier for keeping fish out of an area and excluding animals, which is really, really good. The last thing then that I wanted to talk about in terms of data was to talk about this concept of acclimation. So then an acclimation is basically saying, can fish get used to a high CO2 environment? Which obviously has potential if you're putting in a fish barrier. If you install it and that the animals suddenly become used to it, then they might be able to swim through it, right? So we want to make sure that animals simply don't get used to being in a high carbon dioxide environment. So what we did for this experiment was we collected fish, and we used largemouth bass as a surrogate species. We weren't able to get Asian carp for this, but you see this tank. We put them in a tank, like you see at the very top. That actually is the tank, and you can see the little largemouth bass kind of swimming around inside the tank. Every individual was marked with an individual tag, so we knew basically who everybody in the tank was. We grabbed the animals out of the tank. We ran them through that shuttle box testing again. So you put them in one side, raise the CO2, watch them leave, raise the CO2 in the next side, watch them leave, and ping pong them back and forth, and you get an individual. You can say fish 1 left at this concentration, fish 2 left at this concentration, and we know the concentration where each individual left. After we're done that testing, put everyone back in the tank, leave them in those tanks for 8 weeks, and then in that 8 weeks we raised carbon dioxide concentrations to be pretty high, about as high as we could take it without the animals all passing out. Left them there for 2 months, and after 2 months we pulled them all out and retested them within that shuttle box to see if their avoidance thresholds changed after 8 weeks of sitting in high CO2. And introduced the data are here, so now the y-axis, the vertical axis again, the concentration when animals just chose to move, the control period prior to CO2 on the left, and then on the right is after 8 weeks, 2 months at high CO2. And what you see is that there is no difference in the concentration when animals chose to move. So keeping animals at high CO2 for 2 months does not impact their choice and does not impact whether or not they leave an area and whether or not they decide. There's no impact of acclimation, and CO2 exposure does not change avoidance thresholds. So to start to wrap everything up and bring all these different chunks that I've talked about together, freshwater biodiversity is highly threatened, and we're losing biodiversity from freshwaters at an alarming rate, and invasive species are a major threat and a major part of that problem. Preventing the arrival of invasive species is a much better, much more efficient strategy than trying to remove invasive species after they've established. Hopefully the data that I've shown you today will convince you that carbon dioxide has potential to act as a non-physical barrier to impact fish movements, acting as a push barrier, something to keep fish out of an area and prevent them from moving into new habitat. Acute exposures induce stress responses. We see equilibrium loss, but the big thing is that fish avoid zones of elevated carbon dioxide. This does not appear to be size specific. It impacts juveniles, it impacts adults. It does not appear to be species specific. Largemouth bass, bluegill, carp, they all seem to respond in the same way. but we do need to reach target threshold, likely in the ballpark of 50 to 70 milligrams per liter, or there's a bunch of different ways you can report these data. And so it's called microatmospheres, which is the pressure. You got to get up to about 60,000 microatmospheres. But the point is there needs to be a threshold for you to have a response for the fish. So despite all the benefits that I've shown, there's still a lot of work for us to do to be able to move things forward. We really want to scale this up even larger. So what we want to do is try and build a fish barrier at a very, very large scale. So the top picture there is a test site that we have not too far away from Champaign in a gravel pit. And you can see there's that blob of yellow in the middle is a person in a yellow shirt. And so we're really trying to build a carbon dioxide barrier now at a very, very large scale, just to figure out logistics, cost, how to do this, that kind of stuff. We're trying to deploy carbon dioxide into a flowing water environment. There's a number of different ways where we've thought about deploying carbon dioxide, but one of the options is to have it deployed in a river, in flowing water. Everything we've done really so far has been in a tank or in a pond, and so we want to move into flowing water. We need to quantify impacts on non-target organisms. So again, we're looking at fish that aren't Asian carp, mussels, realizing there's a lot of animals that could be present should we build a CO2 barrier. And how would they respond to the exposure, and then just broadly impacts on the receiving environment. We want to make sure we're not doing more harm than good with this, and making sure that things like sort of low pH, where animals are actually going to be able to deal with some of the changes in pH, deal with the changes in CO2 and carbon dioxide. So looking at non-target impacts, environmental impacts broadly, and making sure we're doing everything right before we actually move it into this scale, this kind of application. So with that, I think I'm going to draw things to a close. I'm happy to take questions. I wanted to mention this has been a big project for the past 4 or 5 years now. I want to mention a lot of the folks, both from my lab at the University of Illinois, the folks from the United States Geological Survey, the Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin, have been very, very helpful. Jason Romine from Western Fisheries Data Center, or Western Fisheries Research Center, and then Illinois Department of Natural Resources have been very, very supportive along the way too, and given us a lot of help. So with that, I think maybe I'll draw things to a close. I'm happy to take questions for a little while, and then happy to do what I can to answer things. Sorry, I got one question that's rolled in. Do people eat these fish was the question, and actually, yeah. So in a lot of places, particularly in Asia, they're a lot more popular for whatever reason. Folks in North America really have not got on side with eating carp. I think it's because there's carp. There's kind of a stigma associated with carp. People have been trying to promote that in terms of a control mechanism and get people eating them. Then hopefully people will target them and try and get rid of them. But for whatever reason in North America, they really have not. It has not caught on for people to eat them. Another question would be another question that came in was cost of this type of barrier, and that's a really good question. So there's to do this. There's it's a complicated question to try and answer because we could do things like say there's a lot of ideas that have been bounced around for deploying a CO2 barrier. One option is a permanent installation or semi-permanent installation, long-term installation at a different site. For example, say at a boat lock, or a shipping lock, or in a canal, or those kinds of things. And that's one option. Another option is just sort of a short-term deployment. So, for example, there's areas that Asian carp like to sort of hang out, or maybe there's we know there's Asian places where Asian carp like to spawn. And we might be able to put a CO2 barrier in place short-term for a period of, like, say, weeks or days, just to stop fish from moving into a preferred habitat to maybe negatively impact reproduction. We might be able to keep them penned in an area to help facilitate harvest. So there's a number of ways that CO2 barriers could be used should things progress to that point. So having said that, then there's cost with that infrastructure, and then there's cost with the cost of the gas. Two things on that too. One is that carbon dioxide is very soluble in water, and so the amount of carbon dioxide that's needed to reach target concentration is actually very low. And so it really is relatively easy to get it up to target concentrations. The reality is if you're talking about a river or a boat lock or something, that's a lot of carbon dioxide, but it is relatively easy. The other thing is what we've been using so far has been waste carbon dioxide that when we've got carbon dioxide from our various suppliers, we're getting waste carbon dioxide that's come from soybean processing plants or oil and gas refineries, and so we're using waste carbon dioxide as part of this in terms of CO2 that we've been using. So to sort of bring that then to sort of completion, cost is difficult to pin down because it's going to depend on receiving environment, the chemistry of the water, and the scale of the application. But so it's a little bit challenging to ask. Another question: what about outlier fish? Would those fish be able to make it through? And so I think what that's getting at is again like non-target fishes. From what we've seen, this is not a species-specific barrier. So once you put a CO2 barrier in place, I think it's going to, from what we can tell right now, it's going to stop all fish from moving through it. That can be a good thing. That can be a bad thing. So therefore, it depends on where you're going to deploy this. How are you going to deploy? Are you going to deploy things? If it gets deployed, does it get deployed long term? Does it get deployed short term? But yeah, as far as we can tell right now, there's no. It's not species specific, and any fish just swim away from it, and they just voluntarily choose to just leave. Next question was concern releasing CO2 into the atmosphere, or is that offset by using waste carbon dioxide? So yeah, that's, in other words, there's a lot of stuff with climate change, and the fact that climate change, one of the big drivers is carbon dioxide gas, and so how does that play into all this? And so that's and that's a really good question. The reality is that some of the carbon dioxide that would be with a deployment like this, some of the carbon dioxide would remain dissolved in the water. Some of it would get taken up by bacteria and phytoplankton and those kinds of things that need carbon dioxide, and then some of it probably would off gas and return to the environment. So some of it would, and in terms of the specifics and the different concentrations, it's hard to define at this point. But as I said, what we've been doing so far is taking carbon dioxide that would have been released into the environment as part of waste, as part of oil and gas refineries or those kinds of things, and actually using it. And so there is some purpose to, some benefit to doing that. Whether or not that continues, if things go to a large scale or those kinds of things, is difficult to say right now. But at least to this point, that's how things have kind of been going so far. And hopefully I'm answering all these questions adequately as well. So what do you say to those people? So the next question that's coming, what do you say to the people that say the carp are going to make it to the Great Lakes regardless of what we do? So why are we spending millions of tax dollars to do this? And I think that's a valid point. I think, so in other words, it's, there are people out there that are saying, well, this is just a matter of time, right? No matter what we do, the animals are going to find their way through. None of these barriers are 100% effective. It is just a matter of time. Why are we bothering with this? And that's a good question. And I really don't have a strong answer for that because there's, yeah, I don't have a strong answer for that. I think my sense though is that, a lot of it is, well, at least we're trying. You know what I mean? If animals do get into the Great Lakes, and they do spread sort of throughout the Great Lakes area and the Great Lakes Basin, that kind of stuff, it's nice to know that at least we tried and we really did everything we could to stop things, and we gave it a good effort. We're learning a lot about a bunch of different technologies. We've made a lot of progress on a lot of invasive controls. The reality is there's a lot of things in the Great Lakes we don't want in the Mississippi Basin as well. So a lot of this technology, not just carbon dioxide barriers, a lot of things about stopping animals from moving from the Great Lakes into the Mississippi Basin. So yeah, there's a chance that that's going to happen, but hopefully it won't. Next question is, if Asian carp do get into the Great Lakes, what is the worst case scenario? Yeah, worst case scenario. Well, so I think that's a good question, and we don't have a lot of data on that, and it's difficult to try and predict, and so it's a little bit of a gray area, which is part of the concern, I think. The two things I think are, or a bunch of different things. One is, could they eat and survive in the Great Lakes? Like, so for example, in Lake Michigan or in Lake Erie, could they even survive there? Is there enough food? I think some of the more recent data say probably, possibly. So they could actually negatively impact the native fish species in the Great Lakes themselves. They could get into some of the tributaries and some of the smaller rivers that feed into the Great Lakes and start impacting animals there. And then, then the reality is it goes beyond, they could go beyond the Great Lakes. They could go into, once you get into the St. Lawrence River, then then you can get into things like New York and some of the East Coast spots. So there's a lot of canals on the eastern part of North America, and there's potential for the animals to spread. So not to be all doom and gloom, and we don't really know that what the worst case scenario is, but it could be, there could be some negative consequences there. So far, not seeing any more. Not seeing any more questions. We may be done for questions. Anything else? Oh, there we go. We got one. The question that came in was, why don't we figure out how to get them to jump out of the water onto shore? Well, that's it. And that actually is a good question. They do jump, obviously, and that can cause a lot of problems. Yeah, I think the jumping thing is actually a big deal, and it's everything from people kind of getting hit, which can obviously cause problems for people. But then there's jump in your boat and break all your fishing rods. So there's a lot of bad things with them jumping. It's tough to to figure out how many animals there are because they're all jumping all over the place. We don't really have a good sense of population sizes and those kinds of things. So yeah, so to get them to jump on the shore, that'd be great if we could do that. But I think that they're a little bit too crafty, I think, for that, and they end up just going back into the water, unfortunately. Next question that came in: how many years have I been in the field? So that's in terms of Asian carp, I'd say probably about 5 years now. We've been playing with Asian carp, but then in terms of just general fish biology, fisheries, fish conservation things, probably creeping up now on 15 years, 17 years or so. I started in college and never kind of left, so and it's been a lot of fun. Any more questions? I think I see one coming in. Well, I'm glad. Hopefully, people enjoyed it. I'm glad. Hopefully, people enjoy what we did, and hopefully, it all made sense. I'm happy. I should have put my email on this last slide, but I mean, I'm assuming these slides might be available. If people have questions or want to contact me or whatever, fire me an email. I'm happy to do what I can to help out and answer questions or pass along any information I can. And if you have access to the slides, there's enough stuff there you can kind of go look up if you're really interested. So. somebody, the question that came in is saying that the fish they're creeped out by the fish, and yeah, if you haven't seen it in person, it's actually really it is a little bit creepy. Is probably a good word when they start jumping, and I've been in some of those areas where all of the pictures where there's like thousands of fish in the air, they all come from a very, very small part of the Illinois River where population densities are very, very high. And I've been there and I've taken classes down there and stuff, and it actually is quite interesting, and it's quite startling in a lot of ways. So yeah, if you haven't seen it, it's something to go experience. Well, if there's no more questions, I just want to say thanks again for everyone coming out, and I'm thanks for the invite, and hopefully this made sense. I'm really happy to share and make this stuff available. So definitely drop me an email if you have questions, and it's been really fun for me. So hopefully people enjoyed it. All right, I'm going to sign off. Definitely drop me an email if there's questions, and thanks again. Everyone have a good night. Bye-bye.